Okay. Hi everyone, and my name is Dr. Zhao Zhendong. I'm in charge of the TransGen's marketing offices. So here, tell you something about our next generation sequencing products. Here we have four different kinds of four parts about our NGS products. First one is for DNA library prep kit. Here we provide TN5 DNA library prep kit for Illumina platform. And here we have three different products. First one is for 5 nanogram, and the other is for 1 nanogram, and the third one is for 50 nanogram DNA. And the other one is the DNA library prep kit is a traditional method for lab prep for the Illumina platform. And the third one is for MGI platform. MGI is coming from China. Okay, the other part is for our RNA sequencing lab prep kit. And here we provide a traditional one and a fast one. The fast one means we combine different steps into one, and then it will save your experiments maybe 1 or 2 hours. The third part is for rRNA depletion kits. When we do the RNA sequencing, the most influenced factor is the rRNA, which maybe contains about 90% of the whole RNAs. So rRNA should have been removed before your RNA lab prep. So in this case, we have three different products. First one is for human, mouse, and the rats. And the other one is for the bacteria. And the third one is for the animals. So it means we can deal with all different kinds of species to do the rRNA depletions. And the last one is for different relatives products. For example, for library amplification supermix, mRNA kits, and the size selection beads or RNA beads. Okay, the basic purpose of library preparation is to prepare the dsDNA fragments with these following structures. Here, this gray one is the DNA insert. This one is the target information we want to know about the whole sequence. And the rest part, such as P5, I5, I7, P7, is the structure which is combined with the platform, such as Illumina platform. I5 and I7 is the specific structure for Illumina platforms. And for maybe MGI platform, there's different, maybe we call it P3 or P5. It's a different structures. So in this case, maybe Illumina platform contains about 70% of the market for the whole world. So we most of our products is for Illumina platform. This one is for TN5 DNA lab prep kit for Illumina platforms. The application about these products is suitable for using a small genomes such as bacteria or other double-stranded DNA virus, or even the PCR products, plasmids, which can also be used for these systems. All these small fragments can be used for Illumina high-throughput sequencing platforms, such as CHARX or such as different kind of platform, all can be used for these products. And here we have to make sure for 50 nanogram, for 1 nanogram, or 5 nanogram, it means the templates, the amount of the templates is different. So we combine those different kinds of enzymes to combine different amount of the DNAs. Okay, this TN5 DNA lab prep kit is for Illumina platform. It's developed for the whole systems for Illumina. And this case is suitable for different initial DNAs. And this case use the transposition techniques which can complete the DNA fragments and the adapter ligation simultaneously within 5 minutes. Illumina has the patent for the transposition fragment systems, but in this case, we used different ligation about this system. We can see here, we use the circular ligation for the whole systems, but the Illumina used the mineralized ligation, so we share different kind of patents. And also these techniques, we also applied the Chinese patent, but unfortunately, maybe it's not suitable for America or Europe. In this instance, the transposition and the digest is also just in 5 minutes to complete the whole systems. And after the purification and amplification, so it means maybe you just need 1.5 hours to complete the whole procedures for your lab prep. Here's the comparison with the competitive products. Here we use the whole genome short fragment libraries were prepared with the 5 nanogram human genomic DNA. And using the prep kit from our companies and from the company I means Illumina. And then we use the sequenced and analyzed the result of the library pick and the yield sequencing data quality, GC contribution, and the correlation shows that TransGen's products performs similarly to the mainstream products. Here we can see the pictures here. The red one is our products, and the blue one is the competitors' products. It shows very similar forms about this image. And the other one is the yield about the library, which shows there's a very similar result here. And also, this is the sequencing data quality. So the Q20 and the Q30, maybe the most important factors about the sequence data quality. So we can see here, the Q20 and the Q30 shows the very similar result here. Also, another very important factor is the depth. Depth about the whole sequencing data means maybe the higher the depth, the more specific the result we get. So we can see here is also the 4.6, maybe the very similar result here. And the GC distribution, when we do the sequencing, the GC distribution shows very important result about the whole data. If there's a GC distribution shows very irregular, it means the data is maybe not so confidential. So in this case, we can see the GC distribution very similar result here. Okay, this one is our traditional one because the TM5 shows a very specific patent for the systems. So maybe in China, we have the patent to protect our products. Maybe in Europe or in United States, the patent do not protect our products. So we also have the very traditional one. So this traditional one, there's no any patent conflict for the whole systems. So in this case, this traditional one is a very classic method for the DNA library prep. And then it can be used for whole genome sequencing, target gene sequencing, exon gene sequencing, and other targets capture sequencing. This target capture sequencing means even you can use the PFU polymerase to amplify the target genes to capture the specific sequencing. You can use these products to build your library for the sequencing later. And also, it can be used for metagenomics sequencing. NGS is a very important part of the application in this area. So it means when we use this one, you can do the NGS for the research. But in this case, there's no TM5, so such as fragmentation, ligation, shows very specific steps for the whole systems. So it means maybe it will cost a lot of more time to build your library. But also because it's a very classic one, so it means it can be suitable for many different type of the templates, such as chip DNA, FFP DNA, can also be used in this classic one. But the TN5 DNA polymerase, TN5 lab prep kit is not suitable for chip DNA and FFP DNA. So it's a little difference between the TN5 method and the classic method. And when we use this classic method, it's suitable for from 1 nanogram to 1 microgram fragment of the DNAs. So it means you can choose a very wide range about the DNA, not the specific, such as 1 nanogram, 5 nanogram, 50 nanogram for TN5 systems. So it means when you use this classic one, it's more convenient for your experiment and procedures. Okay, this is also the result for the whole systems. This result shows the comparison of the library yields of the different initial sample inputs. Here we input the 1 nanogram, 10 nanogram, 100 nanogram, 500 nanogram, and 1 microgram. So it shows very similar result compared with the capital K. K means kappa, means the kappa system. But also we have to focus here with the different initial sample inputs. The circulations is different also. So when we use to put the 1 nanogram, means the initial sample input is very low. So it maybe need 30 cycles for the amplifications. But when we use the 1 microgram for the initial sample inputs, maybe there's 3 cycles is enough for the whole systems. And compared with the capital K kappa systems, we can show our yield about our library shows much more than the kappa K. And also, there's a sequencing data quality. This data quality will show many times later because it's the very important factors for the whole systems. Also, the Q20 and the Q30 shows very similar results compared with K. And also here, we use the different templates. The first one is the blood from human, and then the FFP sample from human, and the rice samples. So we can, dealing with the human samples, also with the plant samples, can also get a very good result here. And also the depth. The depth for human is always 8.0 from these systems. And also, when we use the plant samples, the depth is always up to 10. So this 11 shows the very good result here. Also, this is the comparison sequencing result for the GC distribution. The GC distribution shows we can get very good result in this this two shows very similar results. But in this one, the capital K shows there's a lot of GC distribution in high GC templates. Maybe it's up to 50% of the GC content shows a very bad result for the capital K systems. But when we use the other systems, even in the very high GC content, we show very good results and get very specific information for the systems. And also SNP analyze. SNP analyze usually used for DNA sequencing part. When we do the DNA sequencing, SNP shows very specific information for the genomic DNA systems. For example, when we do such as human, we can use the SNP to recognize different human species. Or when we do the rice, we can use the SNP to recognize different kinds of the target genes, the different phenomena for the whole systems. So in this case, we can get the total number of the SNP. SNP shows very important results. So if someone gets the SNP results is much lower than the other one, it shows it get a very good result. But in this case, also our products, but also the kappa systems, we get a very similar result about the total SNP systems for the whole informations. Okay. When we do the DNA lab prep, but it's a very important component here is the size selection DNA beads. It's very easy to use and suitable for automatic work systems. And this one can do the DNA purification and the size selection. We can use this one to produce specific target gene products to do the sequencing part. Such as this one, we can use, this kit is based on the SPRI systems. And also this adopts the magnetic beads and the buffer systems with unique separation functions, and which is suitable for DNA purification, DNA concentration, and the DNA and the NGS lab constructions. When we use this one, these procedures here, we do the DNA purification, and then when we do the procedures, this one, we do the DNA size selections, but with the same buffer and the whole systems. So there's one product with two different procedures, shows two different results here. And this result shows the DNA purification. So when we use the different DNA inputs, and then we use the size selection piece with the whole buffers, and then we can get the whole different DNAs. And this image is performed by Angelin's high sensitivity DNA chip. The machine is the Angelin's 2100 can perform these systems. We can detect the DNA analysis, the situation about the DNA and RNAs. Okay. And when we use the different procedures for the DNA size selection, you can see here. This red one, it shows the no size selection. It's just the DNA purification, just like this one. So it means it will collect all different sizes of the DNAs. So it's the total DNA here. But when we use the different procedure for different kinds of length of the source fragments, we can get different kind of length of the DNA. So it means when we use the size selection DNA beads, you can get specific from 300 to 400 or 600 to 700. But why will you need these DNA size selections is based on different kinds of platforms, such as Illumina platform. They have different kinds of machines to do the sequencing. When we use the X10 machines, it means the fragment maybe is to 250 to 300. But when we use a different machine, I don't remember the machine's name. It's a very small one, can be used for in the hospital. It's very small one, but the high throughput is very low. But it's very easy for doctors to perform the systems. But in this case, it needs the fragments should be at 400 to 500 fragments. So in this case, you can use the DNA size selection to perform different size of the fragments to suitable for different kinds of machines to do the sequencing part. Okay, the next part is RNA lab prep constructions. When we use the RNA sequence lab prep kit, the first one is the RNA depletion. So I will tell you what is the RNA. When we extract the RNA and we use the embryos electrophoresis, we can see the specific bands about the RNA. Here is the 28S RNA, 18S RNA, and the 5.8 RNA. Usually, these three bands shows very good result about the RNAs, but this RNA is not the mRNA, it's not the messenger RNA. This RNA, these three bands, is the rRNA, means the ribosome RNA. So in this case, the ribosome RNA usually share the very same information about different kinds of peoples or animals. So in this case, maybe the ribosome RNA contains about the 90% or 99% about the whole RNA information. So in this case, if we do not do the RNA depletion, when we get a very large amount about the information, maybe the 99% is just a waste of time. So in this case, we have to make sure that RNA is being depleted before the sequencing, before the RNA lab prep constructions. So in this case, we used our RNA depletions, and we show the result here. So we detect the human, mouse, and the rat different RNAs, and the line 1 shows the RNA original one. And the other one, line 2, shows the treatment with the RNA depletion. So we can see here, line 1 shows a very good result about RNA, and maybe line 2, there's nothing. But exactly, line 2, there's a very slight smears. Maybe we cannot see very clearly. It's very smear band here. This smear band is the mRNA messenger RNA. So the messenger RNA just is maybe less than 10% about the whole RNAs. So in this case, it's very important and very, very difficult to get the information about the messenger RNAs. There is 2 different basic principles for RNA depletions. First one is the ribo-zero information principles. Ribo-zero information means we can use the ribo-zero RNA binds to RNA. It means there's a very small oligos and with a universal sequence nucleotide, which can bind to the RNAs. And then these oligos can be removed from the RNA with the magnetic beads to remove the whole systems. So it means we can use the oligo to attract and bind the RNA, and then we use the magnetic beads to remove the whole systems. So the left is the mRNA in the whole systems. And the Illumina and the Thermo use the ribo-zero principle for their RNA depletion systems. And the other one, our company, TransGen company, NEB, Kappa, Visa, these companies use the RNase-H principles for whole systems. The first one is very similar. We use also the DNA oligos, which can bind specifically by the RNA and not bind with the mRNA. And then we use the RNase-H enzymes. These enzymes will digest the RNA, which is only bind with the DNA. So it means if the RNA is free, is just a single line here in the supernet, it will not be recognized by RNase-H. RNase only digest the RNA which is binding with the DNase. And then we use the DNase 1 to remove the DNA oligos and digest the DNA oligo into the nucleotides. The rest is the mRNA. So in this case, we use the 2 different enzymes to perform the whole systems. Ribo-zero use the magnetic beads to remove the RNAs. So maybe these 2 different principles. Sometimes people say that ribo-zero shows a higher efficiency than the RNase-H. But in our companies, we do a lot of experiments. It shows RNase-H shows a similar result with the ribo-zero systems. Also, ribo-zero systems also be protected by the patent from Illumina. So maybe we have to use the RNase-H system for the RNA depletions. Here is also the result here. We use the NGS 2100 RNA chip to recognize these RNAs. This specific peak about the RNA shows here is the 28 siRNA, 18 siRNA, and 50.8 siRNA. So the ring value about the RNA shows the 9.9. The ring value what is the ring value? When we use this chip, the machine will tell us about the completion about the RNAs. So the highest ring value is 10. So maybe no one can get the 10. So 9.9 is a very high good result about the RNAs. So it shows the RNA is very complete. And then we use the RNA depletion. So we can see here, this is the total RNA. It's a very complete RNA. And when we use the RNA depletion systems, it cannot be detected specifically in this chip. And also, we can get the sequencing data here. When we use the RNA depletion and get the analysis, we can see from the human, mouse, and the rats, the RNA remove the percent about the RNA removal is nearly 99%. So this shows very good result compared with the complete kappa. So in this case, you can use this one to dealing with the different species. And the first kit is suitable for human, mouse, and the rat, because this three is the most widely used for the research. And also we have two different products. First one is for bacteria. So these strains that have been successfully tested in our lab, and also the RNA rate is lower than 5%. So this 13 strains we already have been tested in our lab. And also the other animals can also be used these kits. These species that have been successfully tested in our lab, the RNA rate is lower than 10%. So there's 9 species. So maybe the pig, the chicken, zebrafish, such as this one is also widely used for in the lab. And also there's a little more information about the systems. We provide all different kinds of products for animals, for humans, for mouse and rat, for bacteria, but not for plants. So when you're dealing with the plant RNA, you cannot use these products. But the other one, you can use the mRNA beads, can get the similar results for the plant RNAs. Because the plant RNA also has poly-A in the mRNAs, and the mRNA beads can recognize the poly-A from the mRNA of the plant species, and use the mRNA beads can extract the mRNA from the total RNAs. So it shows different principle for the systems. Maybe later we will develop the new kits for RNA deletion for plants. But now we have to use the mRNA beads for plant tissues. Okay, when we're dealing with RNA depletion and get the messenger RNA, which is whose the information is we focus on. So in this case, we use the RNA lab prep kit for Illumina, and also it's very convenient for people to use. And also get a very high data quality for the systems. Also, the application is very wide about the whole transcriptional sequencing, gene expression analysis, and also the SNV analysis for different purposes of the research. This one is a very traditional products, such as classic one, maybe. So in this case, we can see there's many procedures, about 8 procedures about the whole systems. So maybe it will take a long time, maybe 5 to 6 hours to pursue this whole systems. But just as it's a very classic one, it means its applications is very wide. So you can deal with it with different research purpose. You can use this one. Later, I will introduce a new products for the RNA systems. So we can see the sequencing data quality is also very high. Also, Q20 and Q30 also is a very important factors during the sequence. But in the RNA sequencing, RNA rate is also be recognized here. So in this case, we used a human 1 microliter. We get the RNA rate just in 0.75, 78%. So it shows very good result for the RNA depletions. If this RNA rate is up to 10%, it means this data cannot be used. So it has to be lower than 10% for the whole systems. And the Q20 and the Q30 also shares a very similar result compared with the V company and the N company. V is from the Visa from Chinese company, N is for AEB from the American company. Okay. Total mapped and uniquely mapped is also can be recognized in the RNA sequencing. Because when we use the RNA sequencing, we want to get the very specific gene expression levels and the informations. So total map rate also shows the quality about the sequencing data. Gene expression level and the validation analysis. This is very difficult diagram for the systems. Maybe this one is just for the researchers to dealing with this information. So we just need to know our company's products and compared with the different others comparison companies shows very similar results. That's enough for the specific researchers. They will focus on the specific numbers for different expressions and variations. So in this case, we will do not to analyze the specific meaning for each numbers. This one is fast RNA cell prep library kit. The last one I just talked about is a very classic one. It will take 5 to 6 hours to perform the whole systems. But this one is a fast library preparation systems. It means it just can be perform the whole system in maybe 3 to 4 hours. So it will save you 1 or 2 hours for the whole procedures. The procedure about this system, just the 6 steps, is compared with the RNA fragmentation and this one, second strand synthesis and the repair and the attailing. So it compares the 3 steps into 1 step. So it means it will save 1 or 2 hours from these steps. Okay, these steps. Also, qualities here. Q20, Q30 also shows very similar compared with others. And also RNA rate. Let me see. Oh, it shows different. This is the RNA rate from the other diagram. So it's 0.79%. And this one is the specific numbers about the RNA rates. Okay. And the total mapping rate is also very similar from the other companies. So when you use this fast lab prep kit, you can save your time and it shows the very similar results from others companies, from the comparison companies or our classic ones. We have introduced you about the DNA prep lab and the RNA prep lab. But in this case, when we finish the RNA lab construction, we have to use the library amplification supermix to make sure the yield of the library can suitable for the Illumina platforms. So this supermix can be used with this DNA and RNA lab prep kit. But in this case, we use the specific enzymes for the whole systems. We call the PFU system, PFU fly polymerase for DNAs. So in this case, these enzymes should have very high efficiency for the PCR systems and very low bias for different kinds of GC content. Here we detect from different GC content from 33% to 72% of the different GC content, but it shows a very similar result about the application efficiency. Okay, the last one is our products for the NGS products here. So we have maybe nearly 30 different kinds of products here. So you can see a brochure. I can show you a brochure.不是共享屏幕，继续共享。不是共享。
Okay, now you're on main screen.
Okay. This brochure is for NGS products. So our colleagues tell me that this brochure is already emailed to you, so you can see the more information about the NGS products here, and more results shows in these brochures. Okay. So the last one shows the different products we provide. So all different kind of products for the NGS systems. So you can see more information in this brochure. Okay.